Dear students, welcome to EPG Pathshala. I am Dr. Meena Yadav from Maitri College, University of Delhi. Today, I am going to talk about pituitary hormones. After studying this module, you should be able to understand the functional areas of pituitary, describe the significance of median eminence, know the importance of portal capillary plexus, write about the hormones secreted by anterior pituitary and posterior pituitary, discuss the stimulation for the secretion of pituitary hormones, describe the structure of pituitary hormones, write down the functions of the hormones secreted by pituitary. So, let us start with the pituitary gland. The pituitary gland, also known as hypophysis, is a small round gland which lies in a pocket called as sella turcica of the sphenoid bone, and it is connected to hypothalamus by the infundibulum or hypophyseal stalk. It controls the secretion from pituitary gland and several other endocrine glands in the body. The pituitary gland is actually made up of two parts which are structurally and functionally different. That is the anterior lobe, which is also known as adenohypophysis, and a posterior lobe which is known as neurohypophysis. Both the lobes of pituitary secrete several hormones from the specialized cells within the pituitary or from the nerve endings. The pituitary receives blood supply from the portal hypophyseal vessels, which are also called as portal capillary plexus, which pass through the median eminence, a connecting link between hypothalamus and other peripheral organ systems. Now, let us start with the hormones secreted by the anterior pituitary. The anterior pituitary contains several types of cells which have receptors for specific releasing or inhibiting factors. The releasing factors from hypothalamus integrate the nervous and endocrine systems. Thus, when the releasing and inhibiting factors from hypothalamus reach the anterior pituitary via portal capillary vasculature, they bind to specific cells, thus activating a cascade of events within these cells, leading to the release of anterior pituitary hormones in general circulation for various target organs. The anterior pituitary secretes several hormones which include the 6 important hormones, and they are the luteinizing hormone, abbreviated as LH, adrenocorticotrophic hormone ACTH, thyroid stimulating hormone TSH, follicle stimulating hormone FSH, and the growth hormone GH, and the prolactin, that is PRL. Now, the anterior pituitary hormones may also be called as tropic hormones or endocrinokinetic hormones in function, except prolactin, as they act on other endocrine glands or tissues to stimulate them to secrete active substances. The following sections discuss the structure, release and functions of the various hormones secreted by the anterior pituitary. Now, first let us take ACTH or adrenocorticotrophic hormone. The ACTH is also known as corticotrophin. It is secreted by anterior pituitary and stimulate the production and secretion of hormones from adrenal cortex, especially the glucocorticoids. The mode of action of ACTH. The target cells in adrenal gland for ACTH are adrenocortical cells. When ACTH binds these cells, the binding activates adenyl cyclase in the plasma membrane, which produces CAMP or cyclic AMP. The cyclic AMP activates various pathways that lead to the production of adrenocortical hormones. The pathway also includes activation of protein kinase A, which converts cholesterol to pregnenolone, which is a rate limiting step for the synthesis of adrenocortical hormones. The secretion of ACTH by anterior pituitary is stimulated by corticotrophin releasing hormone or CRH from the hypothalamus. The release of CRH from paraventricular nucleus PVN of hypothalamus is regulated by several factors, which include cardiovascular baroreceptors. The ACTH, once secreted by anterior pituitary, it goes into general circulation and then reaches the adrenal gland and stimulates the secretion of adrenal steroids, that is the glucocorticoids. All types of mental and physical stress can lead to heightened secretion of ACTH, within few minutes and subsequent increased cortisol secretion. However, the cortisol has negative feedback effect on hypothalamus and anterior pituitary. So, when its concentration is too high, it inhibits the secretion of CRH and ACTH from hypothalamus and anterior pituitary respectively. The human ACTH is a small peptide and consists of 39 amino acids. Its molecular weight is 4507 Daltons and it has no carbohydrate groups attached to it. The ACTH secretion in anterior pituitary is accompanied by secretion of few other hormones, as all of them they have a common gene which forms a larger protein, that is a pre-pro hormone which is called as pro-opiomelanocortin or POMC. The other peptides which are secreted along with ACTH are melanocyte stimulating hormone, beta lipotropin, beta endorphins, etc. The POMC gene is expressed in several cells including the corticotroph cells of anterior pituitary, POMC neurons in hypothalamus, dermis cells and lymphoid tissue. The differential processing of POMC transcript gives different protein products. So, in corticotroph cells, the enzyme pro-hormone convertase 1 or PC1 yields ACTH, beta lipotropin and beta endorphin, etc. In hypothalamus, the enzyme PC2 is expressed which produces alpha, beta and gamma MSH, but not ACTH. Now, coming to the next hormone, that is the growth hormone. It is also known as somatotropin or somatotropic hormone. GHRH and GHIH are released into the portal capillary system, from where they go to the anterior pituitary and act on the special cells called as somatotrophs and stimulate or inhibit the secretion of growth hormone. Somatotrophs constitute 30% of the anterior pituitary cells. The target cells of growth hormone are the liver cells with receptors for growth hormone and stimulate the release of insulin-like growth factor 1, that is IGF-1 by liver cells. The growth hormone and IGF-1 together act synergistically for the growth, and any mutation in any one of these genes results in the absence of growth hormone or IGF-1, which causes dwarfism. The oversecretion of growth hormone during development results in another condition called as gigantism, that is extremely large stature. However, if there are abnormal levels of growth hormone after adulthood, it results in conditions like acromegaly, and during adulthood only certain tissues are responsive to growth hormone like nose, jaws, fingers, orbital ridges, knees and elbows. Due to this, there occurs deformities and changes in the appearance of these organs. The growth hormone and IGF-1 are both involved in feedback regulation of these hormones. Growth hormone and IGF-1 both act as a negative feedback for the release of GHRH. Thus, GH stimulates the expression of somatostatin. In the absence of growth hormone or IGF-1, somatostatin expression is down-regulated. The secretion of growth hormone by anterior pituitary is controlled by two important hormones released from hypothalamus as well as other factors. Growth hormone releasing hormone, that is GHRH, which is secreted by hypothalamus, growth hormone inhibiting hormone, that is GHIH or somatostatin, which is secreted by hypothalamus. The release of growth hormone is also influenced by other factors like dopamine, VIP, TRH, neuropeptide Y, motilin, interleukins, etc. Along with these, other factors like glucose levels in blood, levels of fatty acids, starvation, sleep, exercise, stress, fever, anesthesia and puberty also influence the release of growth hormone. A state of hypoglycemia in body stimulates the release of growth hormone, while hyperglycemia inhibits the release of growth hormone. Both these conditions influence the hypothalamus, which in turn regulates the release of growth hormone by either secreting GHRH or GHIH. Growth hormone performs several functions in the body, like the main function of this hormone is to stimulate the body to grow by acting mostly on the skeleton and skeletal muscles. Growth hormone has important role during neonatal and postnatal growth. It is also required for changes during puberty. There are several functions at the cellular level which are dependent on the availability of growth hormone, like growth hormone promotes net amino acid uptake and protein synthesis, increased lipolysis, carbohydrate metabolism, increased glucose uptake and its use. It mobilizes fat from adipose tissue and stimulates the cells to utilize more fat and sparing the carbohydrates for energy purposes. Another function of growth hormone is that it accelerates the breakdown of glycogen stored in liver and thus elevates the level of glucose in the blood. This diabetogenic effect of growth hormone causes hyperglycemia, which is similar to what happens during diabetes mellitus. At macro level, it stimulates the growth of organs like heart and kidney, stimulates the hormone production, role in immunity and skeletal muscle growth. Growth hormone is a 191 amino acid protein and contains 2 disulfide bonds. In humans, there are 5 genes in a cluster on chromosome number 17 that code for growth hormone. The final growth hormone secreted into general circulation by anterior pituitary is a complex molecule which consists of HGH-N, peptides derived from HGH-N with different kinds of post-translational modifications like glycosylation and also a splice variant of HGH-N, which lacks the amino acids 32 to 46. The next hormone is thyroid stimulating hormone, TSH. It is a glycoprotein hormone secreted by anterior pituitary which acts on the thyroid gland to stimulate the cells to secrete thyroid hormones. The thyroid gland is a part of the brain pituitary thyroid axis which regulates the metabolism, protein synthesis and development of the brain. The receptors for thyroid hormones are located in the brain, peripheral and autonomic system and other organs like bone, muscle, pituitary, heart, liver, testes and placenta, etc. The secretion of TSH is controlled by thyrotropin releasing hormone, that is TRH, also called as thyroid stimulating hormone releasing hormone, that is TSHRH, which is released by hypothalamus. TRH is synthesized by specific cells in the paraventricular nucleus of hypothalamus and acts on the thyrotropes in the anterior pituitary. The thyrotropes constitute 10% of the cells of the anterior pituitary. The axons of these neurons terminate in the median eminence, which is highly vascularized, and from here the TRH is taken to the anterior pituitary by portal system and stimulates the release of TSH. The TSH is released in a pulsatile manner which follows a 24-hour cycle. TSH is released into general circulation and acts on its specific receptors on cells of thyroid gland which release thyroid hormones. The two potent thyroid hormones are thyroxine or T4 and triiodothyronine or T3. The T3 and T4, they regulate the levels of TRH and TSH by negative feedback mechanism. Actually, the thyroid hormones, they are released in 2 forms, bound to a binding protein and the free form. The free thyroid hormones, they act on the target tissues and carry out the negative feedback regulation, whereas the bound thyroid hormones remain as a reservoir and can be used whenever required. TSH is involved in several functions within the body, which we will discuss right now. TSH stimulates the thyroid gland to secrete thyroid hormones. The primary function of thyroid hormone is to regulate the basal metabolic rate and other processes that generate calories. If the pituitary is removed, the thyroid gland atrophies and the thyroid functions, they are depleted. However, if the TSH is administered within few minutes, the thyroid gland gets stimulated to secrete the thyroid hormones. If the stimulation of TSH is prolonged, it results in the enlargement of thyroid gland and a condition develops which is known as goiter. When the thyroid hormones reach the target organs, they bind to their receptors on the cells and regulate the levels of ATP produced by mitochondria and also alter the protein synthesis. In adults, The normal levels of thyroid hormones are very important as they are required to maintain the normal metabolic activities, but altered levels of thyroid hormones may have serious implications. The altered level of thyroid hormones in fetus causes permanent abnormalities in the nervous system and also affect the growth and bone development. TSH stimulates various thyroid functions like uptake of iodine and organification, synthesis of iodothyronins and their release and control, the growth of thyroid gland. TSH protects the cells of thyroid gland from apoptosis. TSH is also used by the mammalian immune system, especially by monocytes and lymphocytes, including dendritic cells, macrophages and few naive peripheral T cells. These cells produce TSH during initial stages of immune function. Now, talking about the structure of TSH. TSH is a glycoprotein hormone whose size is approximately 30 kilodalton. It consists of two subunits, alpha subunit and the beta subunit, which are coded by two independent genes. Alpha subunit is coded by a gene on chromosome number 6, while beta subunit is coded by a gene on chromosome number 1. The alpha subunit contains 4 exons and 3 introns, while beta subunit contains 3 exons and 2 introns. Both the subunits are tightly but non-covalently bound. The alpha subunit of TSH is identical to those of LH, FSH and HCG and consists of 92 amino acids in humans, while beta subunit is different in these hormones. The beta subunit is also a glycoprotein, and in TSH it consists of 112 amino acids in humans. The TSH is active for a short duration and has a half-life of about 60 minutes, after which it is degraded primarily in kidney and also in liver. Let us now talk about the gonadotropins. There are two gonadotropins, FSH and LH, and they are secreted by specialized cells in anterior pituitary called as gonadotrophs. Both LH and FSH are glycoproteins and work through activating cyclic AMP messenger system in target cells. Both LH and FSH act on the gonads and stimulate the synthesis and release of sex steroid hormones, especially the estrogens, progestins and androgens. These hormones are released in a pulsatile manner and act on their respective target organs, that is gonads, by binding to the gonadotrophs. Both the hormones are controlled by a negative feedback mechanism. However, ovulation is also controlled by a positive feedback mechanism. The secretion of FSH in males and females is controlled by gonadotropin releasing hormone or follicle stimulating hormone releasing hormone, which are released by the hypothalamus. Both LH and FSH are released in a pulsatile manner along with GnRH, but the release of FSH is also regulated by inhibin B, which is a glycoprotein hormone produced by the gonads, and inhibin B inhibits the release of FSH from the anterior pituitary. The secretion of LH in both males and females is controlled by luteinizing hormone releasing hormone LHRH, which is also known as GnRH, and they are secreted from hypothalamus. The major functions of LH and FSH are: in females, FSH stimulates the development of ova in the ovaries and release of sex hormone or estrogen. In females, LH together with estrogen released from ovary stimulates the ovary to secrete mature ova and also prepares the uterus for implantation of the fertilized ovum. LH also stimulates the formation of a corpus luteum in the ovary, which secretes progesterone and prepares the mammary glands for milk secretion. Actually, in females, there is a phenomenon called as LH surge, which means an acute rise of LH, which triggers the ovulation and development of corpus luteum. In males, LH is also known as interstitial cell stimulating hormone, that is ICSH, and stimulates the interstitial cells of testes to secrete testosterone. LH has a negative feedback regulation on secretion of testosterone from testes. Thus, in testes, LH regulates steroidogenesis while FSH regulates the spermatogenesis. In males, FSH is tropic for Sertoli cells and has as receptors on Sertoli cells in seminiferous tubules, while LH is tropic for Leydig cells. Along with testosterone released by Leydig cells, FSH regulates the process of spermatogenesis. Another hormone secreted by Sertoli cells is inhibin B, which acts on anterior pituitary to inhibit the release of FSH, and probably also acts on the hypothalamus to inhibit the release of GnRH. Now, let us talk about the structure of LH and FSH. Both LH and FSH are glycoprotein molecules of size approximately 28 kilodaltons. They have two subunits, that is alpha subunit and beta subunit. The LH and FSH are members of the same family, which includes TSH and HCG. All these molecules have a common alpha subunit which has highly conserved amino acid sequences, but have different beta subunit which gives them the ability to perform different functions. The beta subunit of LH, FSH, TSH and CG have varying lengths, that is from 111 amino acids to 149 amino acids, and have highly conserved cysteine residues. Each beta subunit has 12 cysteine residues which make 6 intra-disulfide bonds. FSH and LH contain hexoses, that is mannose and galactose, the hexosamines, that is N-acetyl galactosamine and N-acetyl glucosamine, and also methyl pentose ficose and sialic acid. The carbohydrate groups which are attached to these hormones decrease their metabolism, thereby increasing their effectiveness. The half-life of LH is 60 minutes, while that of FSH is 170 minutes. And any mutation in FSH receptors leading to loss of its function causes hypogonadism, while mutations which cause gain of function causes ovarian hyperstimulation syndrome. The next hormone is prolactin. Prolactin is synthesized in specialized cells in pituitary gland which are called as lactotrophs. Dopamine from hypothalamus inhibits the release of prolactin from pituitary. The concentration of dopamine in hypothalamus is high and is more abundantly present in median eminence, where it can be released in the portal system and can easily reach the anterior pituitary. Any kind of drugs that decrease the dopamine levels in portal circulation stimulate the release of more prolactin, while exogenous administration of dopamine inhibits the prolactin secretion. Due to altered dopamine secretion, the level of prolactin secretion may increase, resulting in a condition called as hyperprolactinemia. The secretion of prolactin is controlled by prolactin inhibiting hormone, which is also known as dopamine from hypothalamus, which inhibits the release of prolactin. Other than dopamine, the secretion of prolactin is also controlled by other neurotransmitters like TRH and vasoactive intestinal peptide. There are other factors also which control the release of prolactin like light, sound, smell and stress. Now, the functions of prolactin. Prolactin is a lactogenic hormone in females and stimulates the production of milk by mammary glands. However, the release of milk is controlled by another hormone called as oxytocin from posterior pituitary. Prolactin acts in coordination with other hormones like estrogen, progesterone, corticosteroids, growth hormone, thyroid hormone and insulin during lactation, that is the formation and the release of milk. Age, sex and reproductive status influence the release of prolactin. In females, the secretion of prolactin is more as compared to males due to high levels of estrogen. During menstrual cycles in females, prolactin levels are higher during luteal phase than during follicular phase. In mammary glands, prolactin stimulates the growth of ductal and lobulo-alveolar tissues and production of milk protein casein. The release of prolactin is triggered by the suckling activity by the infant. In ovary, prolactin increases LH receptors in the corpus luteum and progesterone synthesis by corpus luteum. Prolactin also works for osmoregulation in fishes and amphibians, primarily due to prolactin receptors in kidney, gut, skin and urinary bladder apart from the mammary gland. It maintains the water and electrolyte balance and reduces the sodium and water loss from kidney and gut. It also has a role in growth and development, especially in processes like metamorphosis in amphibians. It also affects the lipid, carbohydrate and steroid metabolism. It alters the biochemistry in brain and thus influences the behavior in several species. Along with lactation, prolactin also alters the biochemistry of ovary, testes, uterus, prostate of several mammals. It also regulates the immune system by influencing the thymus, spleen and leukocytes like macrophages and lymphocytes. Talking about the structure of the prolactin, prolactin is a member of the somatotropin family of hormones, and the prolactin gene is similar to the gene for growth hormone and placental lactogen. The gene for prolactin is situated on chromosome number 6 and carries 6 exons and 4 introns. There are many variants of prolactin. Along with the pituitary gland, it is also synthesized within central nervous system, uterus and associated tissues, immune system and mammary gland. There are many types of receptors for prolactin which are similar to growth hormone receptors and are distributed in many organs of the body. This was all about the hormones which are secreted from the anterior pituitary. Let us now start with the hormones which are secreted by the posterior pituitary. The posterior pituitary secretes 2 very important hormones, that is oxytocin, that is O2, OT and arginine vasopressin AVP or antidiuretic hormone, that is ADH. However, these hormones are not synthesized by posterior pituitary. Actually, the posterior pituitary receives terminals of magnocellular axons whose cell bodies lie in the supraoptic and paraventricular nuclei of hypothalamus, and their axons extend through the median eminence and infundibular stalk to the posterior pituitary. These neurons are neurosecretory in nature. All the neurons carrying the oxytocin and vasopressin do not reach the posterior pituitary exclusively. Some of them may bifurcate and also go to the anterior pituitary and affects its function. For example, oxytocin may act on the lactotrophs and help in the release of prolactin, while AVP together with CRH may act on the corticotrophs and regulate the stress responses. The neurons present in the supraoptic and paraventricular nuclei of hypothalamus secrete 2 major neural hormones, that is arginine vasopressin and oxytocin respectively. These are called as neural hormones as they are secreted by nerves into the blood in the posterior lobe of pituitary. Both the posterior lobe hormones, they are synthesized as a large precursor molecule and they remain bound to a protein called as neurophysin when present in the granules within the neuron. Oxytocin is bound to neurophysin 1 while vasopressin remains bound to neurophysin 2. The precursor molecule for vasopressin is called prepropresophysin and contains 19 amino acids in the leader sequence, which is followed by the vasopressin, neurophysin 2 and a glycopeptide. The precursor for oxytocin is called as preprooxyphysin and is similar to vasopressin but is smaller in size and lacks the glycopeptide. The genes for OT and AVP lie on chromosome 20 in humans. The precursor molecules are synthesized by the ribosomes within the cell bodies in the supraoptic and paraventricular nuclei. After this, the leader sequences are removed in the endoplasmic reticulum, and when they pass through the Golgi apparatus, they are packaged into secretory granules. These granules pass down through the axons to the nerve endings in the posterior pituitary. These granules are called as herring bodies and they can be easily stained and observed under the microscope. Cleavage of precursor molecule occurs while they pass down the axon, and only the hormones and neurophysins they reach the nerve endings. Now talking about the functions of oxytocin. Oxytocin is a peptide which is synthesized in the paraventricular nucleus. The main functions of oxytocin are: in females, oxytocin acts on the uterine wall by targeting their smooth muscles. During childbirth, the smooth muscles of the pregnant female contract in response to oxytocin. During childbirth, the stimulus to the hypothalamus originates due to distension of the uterus. The number of OT receptors increases 100 times in myometrium and endometrium during labor. Due to increase in the OT receptors, uterus becomes susceptible to plasma OT concentration, and this helps in the distension. Oxytocin stimulates the uterine contractions in two ways. Firstly, by acting directly on the uterine smooth muscle cells, and secondly, by promoting the synthesis of prostaglandins in the decidua, which further enhances the uterine contractions by OT. Similar to this, the smooth muscles of the mammary glands, they contract under the effect of OT and thus they release the milk. The stimulus to hypothalamus for milk ejection originates due to the suckling sensation the mother receives. OT also maintains the secretion of milk by stimulating the prolactin secretion. OT along with AVP plays important role in other social behaviors which are associated with successful reproduction and bonding in pairs. Oxytocin has been found in the hypothalamus and pituitary of males as well, which suggests that some more functions of this peptide exist. There are some more stimulations for OT release and various other functions in both males and females. These stimulations include gastric distension, plasma hyperosmolality, CCK administration, and any medication that may decrease the food intake in the rats. If the plasma concentration of OT is high, there is decrease in the food intake as is seen in the rats. Vasopressin is also called as antidiuretic hormone or ADH, which prevents the excessive urine output as it regulates the salt and water within the body. ADH stimulates the kidney tubules to reabsorb more water and allowing only the concentrated urine to flow out of kidney, which is less in volume, by affecting the behavioral responses for drinking and salt uptake. This causes the retention of water and osmotic pressure of the body fluids increases. In the absence of ADH, the urine becomes dilute and the urine output increases almost 10 times and the osmolality of the body fluids also increases. Other functions of AVP include acting as a vasoconstrictor during hemorrhage. AVP binds to V1A receptors on the smooth muscle cells and induces vasoconstriction. It also stimulates CRH action on corticotrophs in anterior pituitary via V1B receptors. AVP also regulates the stress responses. The release of ADH, it depends on the osmotic pressure of the blood, which is monitored by osmoreceptors, which are situated in hypothalamus, and these receptors include atrial baroreceptors, chemoreceptors and somatic efferents. These osmoreceptors stimulate the neurosecretory cells of supraoptic nucleus to release ADH. There are several stimulators for secretion of ADH like dehydration, pain, stress, trauma, anxiety, acetylcholine, nicotine, and even the drugs like morphine, tranquilizers and some anesthetics. So, let us now summarize this chapter about the pituitary hormones. The pituitary gland, also known as hypophysis, is a small round gland which lies in a pocket called sella turcica of the sphenoid bone, and it is connected to hypothalamus by the infundibulum or hypophyseal stalk. The pituitary gland is actually made up of two parts which are structurally and functionally different, that is the anterior lobe known as adenohypophysis and the posterior lobe known as neurohypophysis. The pituitary receives blood supply from the portal hypophyseal vessels, which are also called as portal capillary plexus, which pass through the median eminence and which is a connecting link between hypothalamus and the other peripheral organ systems. The anterior pituitary secretes several hormones which include luteinizing hormone, adrenocorticotrophic hormone, thyroid stimulating hormone, follicle stimulating hormone, growth hormone, and prolactin. ACTH stimulates secretion of glucocorticoid hormones from adrenal gland. The human ACTH is a small peptide and consists of only 39 amino acids. Its molecular weight is 4507 daltons and it has no carbohydrate groups attached to it. The secretion of ACTH is stimulated by corticotrophin releasing hormone CRH from hypothalamus. Growth hormone is also known as somatotropin or somatotropic hormone. The main function of this hormone is to stimulate body to grow by acting mostly on skeleton and skeletal muscles. The secretion of growth hormone is controlled by two hormones, that is which are released from hypothalamus, and they are known as growth hormone releasing hormone GHRH and growth hormone inhibiting hormone, that is GHIH, also known as somatostatin. The target cells of growth hormone are liver cells with receptors for growth hormone and stimulate the release of insulin like growth factor 1 by liver cells. The growth hormone and IGF-1 together act synergistically for the growth, and any mutation in any one of these genes results in the absence of GH or IGF-1, and this causes dwarfism. However, over secretion of growth hormone during development results in gigantism, that is extremely large stature. Growth hormone is a 191 amino acid protein and it contains 2 disulfide bonds. There are 5 genes in a cluster on chromosome number 17 that code for growth hormone. Thyroid stimulating hormone is a hormone secreted by anterior pituitary which acts on the thyroid gland to stimulate the cells to secrete thyroid hormones. The secretion of TSH is controlled by thyrotropin releasing hormone, that is TRH, from hypothalamus. TSH is a glycoprotein hormone whose size is approximately 30 kilodalton. It consists of 2 subunits, alpha subunit and beta subunit, which are coded by 2 independent genes. The alpha subunit gene lies on chromosome number 6 while beta subunit gene lies on chromosome number 1. The alpha subunit contains 4 exons and 3 introns while beta subunit contains 3 exons and 2 introns. FSH and LH, they are called as gonadotropin hormones and they are secreted by specialized cells in the anterior pituitary called as gonadotrophs. Both LH and FSH are glycoproteins and they work through activating CAMP messenger system in target cells. The secretion of FSH and LH in males and females is controlled by gonadotropin releasing hormone, that is GnRH. In females, FSH stimulates the development of ova in the ovaries and the release of sex hormones or estrogen. In males, LH is also known as interstitial cell stimulating hormone ICSH and stimulates the interstitial cells of testes to secrete testosterone. In males, FSH is tropic for Sertoli cells and has receptors on the Sertoli cells in seminiferous tubules, while LH is tropic for Leydig cells. A hormone secreted by Sertoli cells, which is known as inhibin B, acts on the anterior pituitary to inhibit the release of FSH. Both LH and FSH are glycoprotein molecules of size approximately 28 kilodaltons. They have 2 subunits, alpha subunit and beta subunit. Prolactin is synthesized in specialized cells in pituitary gland which are known as lactotrophs. Dopamine from hypothalamus inhibits the release of prolactin from pituitary. The secretion of prolactin is controlled by dopamine from the hypothalamus, which inhibits the release of prolactin. Other than dopamine, the secretion of prolactin is also controlled by other neurotransmitters like TRH and vasoactive intestinal peptide. Prolactin is a lactogenic hormone in females and stimulates the production of milk by mammary glands. It increases the LH receptors in the corpus luteum, works for osmoregulation in fishes and amphibians, has role in growth and development, especially in processes like metamorphosis in amphibians. The size of prolactin molecule is 23 to 24 kilodaltons, that is 199 amino acids, and it contains 3 disulfide bridges. The posterior pituitary secretes 2 important hormones, oxytocin and arginine vasopressin or antidiuretic hormone from paraventricular nucleus and supraoptic nuclei respectively. These hormones remain bound to a protein called as neurophysin when present in the granules within the neuron. Oxytocin is bound to neurophysin 1 while vasopressin remains bound to neurophysin 2. The gene for OT and AVP lie on chromosome number 20. The precursor molecules for vasopressin is called pre-pro-presophysin and contains 19 amino acids in the leader sequence, followed by vasopressin, neurophysin 2 and a glycopeptide. The precursor for oxytocin is called pre-pro-oxyphysin and is similar to vasopressin but is smaller in size as it lacks the glycopeptide. In females, oxytocin acts on the uterine wall by targeting their smooth muscles during childbirth. The smooth muscles of mammary glands also contract under the effect of OT and thus release the milk. The stimulus to hypothalamus for milk ejection originates due to suckling sensation the mother receives. Vasopressin is also called as antidiuretic hormone, which prevents the excessive urine output as it regulates the salt and water within the body. It also acts as a vasoconstrictor during hemorrhage and regulates the stress responses. The release of ADH depends on the osmotic pressure of the blood, which is monitored by osmoreceptors which are situated in hypothalamus like atrial, baroreceptors, chemoreceptors and somatic efferents. These osmoreceptors stimulate the neurosecretory cells of supraoptic nucleus to release ADH. Thank you.